decision making. Finally, people whose corpus callosum is completely sectioned have two entirely separate conscious portions of the brain. For example, in a teenage boy with a sectioned corpus callosum, only the left half of the brain could understand both the written word and the spoken word because the left side was the dominant hemisphere. Conversely, the right side of the brain could understand the written word, but not the spoken word. The two halves of the brain have independent capabilities for consciousness, memory storage, communication, and control of motor activities. The corpus callosum is required for the two sides to operate cooperatively at the superficial subconscious level. The interior commissure plays an important role to unite the emotional responses of the two sides of the brain. What are thoughts, consciousness, memory? Destruction of large portions of cerebral cortex does not prevent a person from having thoughts, but it does reduce the depth of thoughts and also the degree of awareness of the surroundings. Now let's talk about the definition of thought in terms of neural activity as follows. A thought results from a pattern of stimulation of many parts of nervous system at the same time, involving cerebral cortex, thalamus, limbic system, upper reticular formation of the brainstem, and this is known as the holistic theory of thoughts. The stimulated areas of limbic system, thalamus, reticular formation are believed to determine the general nature of thought, giving it such qualities as pleasure, displeasure, pain, comfort, crude modalities of sensation, localization to gross areas of the body, and other general characteristics. Specific stimulated areas of cerebral cortex, they determine discrete characteristics of the thought, such as specific localization of sensations on the surface of the body and of objects in the fields of vision, the feeling of texture of self, visual recognition of rectangular pattern of a concrete block wall. Consciousness can be described as continuing stream of awareness of one's surroundings. Now, memory roles of synaptic facilitation and synaptic inhibition. Memories are stored in the brain by changing the basic sensitivity of synaptic transmission between neurons as a result of previous neural activity. The new or facilitated pathways are called memory traces. They are important because once the traces are established, they can be selectively activated by the thinking mind to reproduce the memories. Memory traces can occur at all levels of the nervous system. Also, long-term memories result from changed synaptic conduction in lower brain centers. Memory associated with intellectual processes is based on memory traces in the cerebral cortex. Positive and negative memory sensitization or habituation of synaptic transmission. Now, we often think of memories as being positive recollections of previous thoughts or experiences. Probably the greatest share of our memories is negative, not positive. If our minds attempted to remember all this information, the memory capacity of the brain would be rapidly exceeded. The brain has the capability to learn to ignore information that is of no consequence. This results from inhibition of synaptic pathways for this type of information, which results in the effect known as habituation. This type of negative memory. Conversely, for incoming information that causes important consequences, such as pain or pleasure, the brain has a different automatic capability of enhancing and storing the memory traces, which is known as positive memory. And it results from facilitation of the synaptic pathways, and the process is called memory sensitization. Now we should be knowing about the classification of memories. Some of the memories they last for only few seconds, some for hours, some for days, months, or even years. Now classification of memory is into three, which is short-term, intermediate long-term, and long-term memory. Now let's talk about them one by one. The first one is the short-term memory. Now, a short-term memory includes memories that last for seconds or at most minutes unless they are converted into longer-term memories. Intermediate long-term memories, they last for days to weeks, but then later they are going to fade away. Then long-term memory, which once stored can be recalled up to years or even a lifetime later. Now these classifications divides memory into declarative memory and skill memory. Declarative memory basically means memory of the various details of an integrated thought, such as memory of an important experience, which includes the memory of surroundings, of time relationships, causes of experience, memory of meaning of the experience, memory of one's deductions that were left in the person's mind. Skill memory, it is frequently associated with motor activities of the person's body, such as all the skills developed for hitting a tennis ball, including automatic memories to sight the ball, calculate the relationship and speed of ball to the racket, and to deduce rapidly the motions of the body, the arms, and the racket required to hit the ball as desired. All of these activated instantly based on previous learning of the game of tennis, then moving on to the next stroke of the game while forgetting the details of the previous stroke. Short-term memory is typified by one's memory of 7 to 10 numerals in a telephone number, or 7 to 10 other discrete facts for a few seconds to a few minutes at a time, but lasting only as long as the person continues to think about the numbers or the facts. This short-term memory is caused by continual neural activity resulting from the nerve signals. Now, short-term memory is presynaptic facilitation or inhibition. This occurs at synapses which lie on terminal nerve fibrils immediately before these fibrils synapse with neuron. The neurotransmitter chemicals secreted at such terminals frequently cause facilitation or inhibition, which is lasting for seconds up to several minutes. Circuits of this type could lead to short-term memory. Intermediate long-term memory. Intermediate long-term memories may last for many minutes or even weeks. They will eventually be lost unless the memory traces are activated enough to become more permanent. Then they are classified as long-term memories. Memories of the intermediate long-term type can result from temporary chemical or physical changes or both in either the synapse presynaptic terminals or the synapse postsynaptic membrane. Changes that can persist for a few minutes up to several weeks. Now, the memory based on chemical changes in the presynaptic terminal or postsynaptic neuronal memory. When the sensory terminal is stimulated repeatedly, but without stimulation of the facilitator terminal, signal transmission at first is great, but it becomes less and less intense with repeated stimulation until transmission almost ceases. This phenomena is habituation. It is a type of negative memory that causes the neuronal circuit to lose its response to repeated events which are insignificant. Conversely, if a noxious stimulus excites the facilitator terminal at the same time that the sensory terminal is stimulated, then instead of transmitted signal into the postsynaptic neuron becoming progressively weaker, the ease of transmission becomes stronger and stronger, and it will remain strong for minutes, hours, days, or with more intense training up to 3 weeks even, without further stimulation of the facilitator terminal. Thus, the noxious stimulus causes the memory pathway through the sensory terminal to become facilitated for days or weeks thereafter. It is especially interesting that even after habituation has occurred, this pathway can be converted back to a facilitated pathway with only a few noxious stimuli. Now let's talk about the molecular mechanism of intermediate memory. Now, mechanism for habituation at the molecular level. The habituation effect in the sensory terminal results from progressive closure of calcium channels through the terminal membrane, though the cause of this calcium channel closure is not fully known. Nevertheless, much smaller than normal amounts of calcium ions can diffuse into the habituated terminal, and much less sensory terminal transmitter is therefore released because calcium entry is the principal stimulus for transmitter release. Then the mechanism of facilitation. In the case of facilitation, at least part of molecular mechanism is believed to be the following one. That is, the stimulation of facilitator presynaptic terminal at the same time that sensory terminal is stimulated causes serotonin release at the facilitator synapse on the surface of sensory terminal. The serotonin acts on serotonin receptors in the sensory terminal membrane, and these receptors activate the enzyme adenyl cyclase inside the membrane. The adenyl cyclase then causes formation of cyclic adenosine monophosphate, that is CAMP, inside the sensory presynaptic terminal. The cyclic AMP activates protein kinase, causing phosphorylation of a protein which itself is part of potassium channels in sensory synaptic terminal membrane, and is going to block the potassium conductance. The blockage can last for minutes up to several weeks even. Now, the lack of potassium